Hello, my presentation is on heat shock proteins in organisms for stress tolerance. So the role of heat shock proteins is that it's a primary moderator of cell stress, and it allows for organisms to adapt to harsh environments. And the three major roles of the heat shock protein is that it upregulates in stressful conditions. So there's higher production of heat shock proteins when there's exposure to environmental stress like infection, inflammation, and high temperature heat shock. And it could also be a chaperone where the heat shock protein functions as intracellular chaperones for other proteins, and it's important in folding and assisting establishment of proper conformation. And it also is a cell monitor where it occurs under non-stressful conditions, and it just monitors cells proteins, and it acts like a proteasome to carry old protein out from cells. And so I'm going to go over the quick history of the discovery of the HSP. So the HSP was in 1962, Italian geneticist Ferruccio Virtuoso reported that heat and the metabolic uncoupler 2,4-dinitrophenol induced a characteristic pattern of puffing in the chromosomes of Drosophila. This discovery eventually led to the identification of the heat shock proteins, or stress proteins, whose expression this puffing represented. So this increased synthesis of selected proteins in Drosophila cells following such stresses from heat shocks were reported first in 1974. And so in 1974, researchers Tissieres, Mitchell, and Tracy discovered that heat shock induces the production of a small number of proteins, and this inhibits the production of most others. And so this research gave rise to a large number of other studies on the induction of heat shock and its biological roles. And yeah, so that's how there's been more research on HSP nowadays. And so the important HSPs I'm going to be talking about are the HSP60, HSP70, and HSP90. There are a lot more HSPs that are there, but these are the major ones. And so HSP60 is also known as chaperones, is a family of heat shock proteins originally sorted by their 60 kDa molecular mass. They prevent misfolding of proteins during stressful situations such as high heat by assisting protein folding. HSP60 belongs to a large class of molecules that assist protein folding called molecular chaperones. And so these are proteins that usually fold from a linear chain of amino acids into a three-dimensional tertiary structure. The next one is HSP70, which is it's a protein with similar structure existing in virtually all living organisms. And so it's intracellularly localized, and it's an important part of the cell's machinery for protein folding, performing chaperoning functions, and helping to protect cells from adverse effects of physiological stresses. Additionally, membrane-bound HSP70s have been identified as a potential target for cancer therapies, and their extracellularly localized counterparts have been identified as having both membrane-bound and membrane-free structures. And then HSP90 is a chaperone protein that assists other proteins to fold properly, stabilizes proteins against heat stress, and aids in protein degradation. It also stabilizes a number of proteins required for tumor growth, which is why HSP90 inhibitors are investigated as anti-cancer drugs. And so we could see that HSPs all sort of have like a similar role of protecting and regulating cells. So I'm going to be talking about the technical advancements that have led to deeper research in HSP, and I think this is an important part because without understanding the technology, then we don't understand where a lot of this information came from. And the first one is omic quantification of nucleic acids and proteins. Transcriptomics has revealed the complexity of expression patterns and has been extensively applied to wild populations. Advances in whole genome sequencing simplified detection of HSP genes and duplication of them. And so such advances has allowed us to construct a consensus phylogenetic tree of HSP70 superfamily members. And from it, we could demonstrate that HSP70 genes have undergone a remarkable expansion. In this particular figure, it's on the oyster Crassostrea gigas, an intertidal species prone to natural thermal stress. From sequencing 61 transcriptomes from the C gigas, it's subjected to 9 stressors, and they showed that the expansion in copy number is correlated with an extraordinary upregulation of HSP70 compared with all the other transcripts. In this case, knowledge at both the genomic and transcriptomic levels has benefited for these researches. And another example is mass spectrometry-based proteomics, which has helped further expand on this research. And so this technique was applied to freshwater versus brackish water spawning of the European whitefish Coriogonus avaritus. With it, they discovered that HSP90 holds a central position in an interaction network in freshwater whitefish, whereas both HSP70 and HSP90 do in the brackish water whitefish. And so, in other aspects, the brackish and freshwater protein networks differ considerably, and when combined with complementary molecular and cellular techniques, MS-based proteomics provides a framework for translating large datasets into an understanding of complex biological processes and phenotypes. And so, previously the ability to manipulate specific genes and their products was experimentally and it was only restricted to lab settings. But this restriction is far less because of RNA interference. And so RNA interference of HSP70 and HSP90 showed us a causal relationship between expression and dehydration tolerance for a mosquito species, and suppressing expression of HSP23 and HSP70 in the flesh fly Sarcophaga crassipalpus with RNAi profoundly affected temperature tolerance. So I'm going to be briefly talking about the evolution of heat shock proteins and how these proteins evolve. So epigenetic changes in HSPS can be mediated by DNA 5 cytosine methylation or RNA 6 adenosine methylation, and this may cause transgenerational effects. Regulatory/coding mutations associated with HSPS include cisgenetic changes caused by insertion or deletion in the regulatory or coding region, and transposable elements, for example, P elements, are a common agent for insertional mutation. Genomic changes of HSPS such as gene copy expansion, gene loss, or pseudogenization can alter HSP expression as well. These mechanisms can drive the evolution of HSP expression at different evolutionary scales. So I'm going to be talking about transposable elements, and these are a potent source of evolutionary variation in HSPS. There are screens of 18 heat shock genes and 48 natural populations of Drosophila, and it showed that the proximal promoters of these heat shock genes harbor more than 200 distinctive transposable elements in 42 of the populations. And these transposable elements vary in size, orientation, insertion site, and allele frequency. And so by changing the transposable elements, we could change the source of HSP. In Drosophila's HSP70, for example, greater than 96% of transposable elements are P elements. Insertion of transposable elements into promoter of HSPs can change HSP expression and in turn variations in stress tolerance, fecundity, and development. And methylation. Epigenetic variations such as methylation can also clearly affect HSP expression, but we don't really know the evolutionary adaptations of it yet. And so, still to be resolved is the persistence of epigenetic variants of HSPs and evolutionary time. Evolutionary variation seems to occur on different time scales in most cases, but regulatory coding, genetic changes, indels, and SNPs in regulatory region that modify gene expression can be on a microevolutionary scale and genomic changes on macroevolutionary scales. So now I'm going to be talking about some different species that express HSP kind of in a unique way. So what happens when HSP is expressed? When HSP is expressed, it can reduce the number of proteins damaged due to heat or proteotoxic stress. This leads to cancer when protein synthesis process leads to error. HSP levels increase when cells undergo heat shock and other stress, such as osmotic stress, salt, microgravity, UV radiation, and light. HSP genes are expressed normally to protein fold, maintain signal transduction, and to proceed with normal development of organisms. HSP levels decrease when heat shocks are absent. Therefore, HSP whether the organism is dealing with natural or abnormal stress levels. So a good example of this is the Belgica antarctica HSP expression. So HSP 90 and HSP 70 and small HSP are expressed at optimal temperature of 4 degrees Celsius. However, in the larvae form, it showed that the HSP expression levels remained the same whether the temperature was increased or decreased to suboptimal levels. So here we're seeing a northern blot hybridization of HSP expression of the Belgica antarctica at 4 degrees Celsius. At 0 to negative 5 degrees Celsius, there is no upregulation in the HSP gene. Even when the larvae returns to optimal temperature, there is no upregulation of the gene. And at 30 to 35 degrees Celsius, there's a loss of signal, meaning the larvae most likely died. This figure illustrates the Belgica antarctica larvae does not respond to temperature changes by upregulating HSP expression. However, adult Belgica antarctica increases HSP expression when temperatures increase. This difference is most likely due to the different niche. So the larva form of the Belgica antarctica spends 2 years in a thermally stable environment, which makes them not have to deal with stress. Therefore, they don't have a reason to express HSP. However, the adult Belgica antarctica, while it only lives a couple of weeks during the summer, they experience a lot of temperature changes and a variety of environments. So they sleep in a thermostable environment at optimal temperature, but then they have to come out into high heat to search for food and material. So therefore, it makes it necessary for the adults to express the HSPs. um, so another example that is kind of unique is the Locusta migratoria. And so HSP expression levels are not always linked to direct evolutionary adaptations, and we need to think about some alternatives. Locusta migratories are known for forming migratory swarms when there is a food shortage or any unfavorable condition. And so when these species are crowded together, the HSPs are upregulated. And so what researchers found was that the HSP expression levels were a lot higher when these locustas, they were in crowded or gregarious situations. So the reason for this is because when they are in a crowded or gregarious situation, there is more vulnerability to disease. And so in order to prevent disease from destroying the organism, HSP um, evolutionary evolutionarily adapted to expressing at higher levels in order to prevent the disease from spreading. And so we could see that in the 6 HSP genes the researchers tested, it was mostly high at the crowded and gregarious levels. And so another interesting thing is that in normal Locusta population, locusts at latitude 18 degrees are more heat tolerant than the locusts at latitude 41 degrees. The difference in the latitude leads to difference in expression levels of HSP70 and HSP90. HSP90 is expressed at higher latitudes, while HSP70 is at lower latitudes. Therefore, we can conclude that HSP expression plasticity may have evolved in order to deal with different temperatures at different latitudes. In the recent research, there has been discovery that HSP90 coding region in the Locusta migratoria is still mutating, and mutant embryos grow up much more quickly than the ones without the mutation. However, this leads to lesser population variation in the developmental rate. And so this figure is a research that provides evidence that HSP expression helps the organism to be able to quickly adapt to its environmental change that leads to low genetic variation. Natural selection acts on HSP90, which reduces genetic variability at HSP90 as well as phenotypic variation. And an incomplete selective sweep occurs in HSP90 mutants linked with an insertion of a transposable element in the HSP90 coding region in a natural locust population. Compared with the wild type, the mutants present faster embryonic development, but reduced within population variation in the developmental rate. That is, high developmental synchrony of embryos dictates segregating sites flanking the transposable element insertion. And so, to wrap up this presentation, I have some feature studies. And one of the feature studies with HSP is exploring cytoprotective and immunoregulatory roles of HSPs. And so we know that the HSPs are divided into 5 major families: the HSP90, HSP70, HSP60, and HSP100. And experimental studies have proved that overexpression and inhibition of HSPs play an important role in maintaining the tolerance and cell viability under above described stress conditions. HSP90 is found to be a promising candidate for the diagnosis, prognosis, and treatment of cancer. Similarly, HSP70, HSP60, and small HSPs experimentally and clinically have potential for the treatment of neurodegenerative disease, ischemia, cell death, autoimmunity, and graft rejection. So HSPs can be a new way for the drug discovery and treatment of critical diseases. And recent evidence indicates that connections between HSPs and cellular machinery in different disease states. So collectively, the studies suggest that HSPs can be used as novel molecular targets for both the pharmacological and therapeutic interventions to prevent various diseases. With the effort of dedicated laboratories, there should be optimism about the rapid development of novel chemical therapies for unfolding/misfolding diseases based on function of the core domain of HSPs. An understanding of different sites for subunit-to-subunit interaction, target substrate protein binding, phosphorylation and interaction with cytoskeletal elements, small metabolites, pharmaceuticals, and nucleotides. We could continue the research that is needed to define the physiological and biological function of all the HSPs identified to date. Thank you, and here are my sources.